So thanks, Shahid. And again, I also want to thank Ashoka University for this opportunity. And indeed, I look forward to interacting with Ashoka University on my visits to India, which hopefully will resume soon. So what I'm going to do today is talk to you about some of our recent unpublished data on immune memory to COVID-19 in the convalescent patients. And my talk today is going to be divided in three parts. In the first part, I'll just give you a brief overview of immune memory, a little bit of a historical perspective on protective immunity, and some general slides about principles of immunological memory. Since this is a broad audience and not everyone is an immunologist, so this will be very basic information about what really immunological memory is all about. Okay. I will then go into the main data that I'll show in this talk. And as I mentioned to you, almost all of this is unpublished data at the moment. A preprint is available and our paper is currently in. I'll then make a few comments about implications of what we have found for protective immunity. And then I don't have that bullet point listed on this slide, but I'll end by saying a few words. Actually, I'll finish this part of the talk, but then I'll end by saying a few words about the collaboration that the Emory Vaccine Center has had at ICGEB for many years, and some of the work our group at ICGEB is doing on COVID-19. Just a few comments with a couple of slides. Okay, so let me start with some comments about immunological memory. Observations of immunological memory, although it wasn't known at that time, even about the immune system or about pathogens, go back actually to several thousand years back to the time of the 3,000-4,000 years back in Greece, where the writers there had noted that people who survived the plague did not get the plague again. Most people died from the plague, but the ones who survived did not get it again. Of course, at this time, microorganisms had not been discovered. The cause of any infectious disease was not discovered, nor was there any knowledge about the immune system. And then over the years, there have been repeated observations of this in the literature. People who get smallpox usually took care of people who survived smallpox. Smallpox had a very high mortality rate prior to the development of the vaccine. And the ones who survived actually took care of people who got smallpox the next time, and they were really not in much danger of getting it. So many, many infectious diseases observations have been made. But what I want to share with you today is a really beautiful experiment of nature that took place on a remote island in Europe. And this experiment of nature took place at the Faroe Islands, which, as you can see from this map, are very isolated. They're south of Iceland and pretty far from the mainland Europe. And this experiment happened or I should say started, in 1781, when there was a measles epidemic on the Faroe Islands. At this time, measles was very widespread all over the world. And again, measles is a terrible disease, can kill. Mortality rates can be, yeah, hello? Yeah, mortality rates can be very high. So at that time, many people in the world were dying, and they also had a major epidemic. But then after that epidemic in 1781, due to some political reasons, there was blockade of these Faroe Islands from transport and communication and interaction with the mainland of Europe. At this time, Faroe Islands were under the Danish government. There was some disagreement. And so the Faroe Islands remained very isolated. There was really no contact with people from the mainland of Europe. And this resulted in the Faroe Islands kind of being protected from more measles coming onto the island. And these restrictions were then removed in 1845. So there was like a 50, 60-year period where there was no commerce between the mainland of, um, of Europe, these isolated Faroe Islands. And then regular commerce started in 1845. This resulted in a major epidemic of measles in 1846. So this is about almost 75 years later, this epidemic happened. And this epidemic was so vast that 75 to 95% of the population got measles. So there was a Danish physician, actually a medical student at that time, Peter Ludwig Panem, who was in medical school. He was 26 years old at that time and a medical student. He was doing a thesis, our modern day MD PhD type of student. And he went and wrote a remarkable paper. Anyone with any interest in infectious diseases or epidemiology, I would strongly recommend reading his paper, which actually was his thesis. And then he made, and you can find this thing actually in this medical classics over here. And in this observation of this epidemic, Ludwig Panem made many, many interesting observations, but I quote here two of them, which really provided remarkable insight into protective immunity. So what Panem wrote was that of the many aged people still living on the Faroes who had had measles in 1781, that is 65 years earlier, okay, not one was attacked the second time. So there were some old people there who had gotten measles earlier when that first this thing had happened, which will take you back. They had gotten it in 1781. And none of these people, older people, got measles second time. And then he had a very nice control group. All the old people who had not gone through with measles in earlier life were attacked when they were exposed to infection. These words I've taken exactly from, I've not modified this. This is exactly what Panem wrote in his thesis. So this was a wonderful control. And it showed that if they had not gotten measles 65 years earlier, the old people were also highly susceptible, just like the younger people were. But if they had gotten measles 65 years earlier, those people were protected. So what this showed for the first time is that re-exposure to the pathogen was not essential to maintain long-term immunological memory. That there were intrinsic mechanisms within that individual, intrinsic immune responses that we now know what immune responses are, immune responses that really were responsible for this protection. So protective immunity for 65 years in the absence of re-exposure. Again, I highly recommend reading this thing that actually Panem published in 1847, his thesis, and then is reprinted in this medical classics volume. So how do we get protective immunity for long term? How do we maintain it? So I'll spend the next 5 minutes or so just telling you about the main players in this protective immunity. So the first line of defense is pre-existing antibody. That is antibody that was induced either upon vaccination or upon infection when one first gets infected, and then there is still pre-existing antibody in the serum and probably also in other places of the body, but certainly can be detected in the very easily can be measured in the serum. So this pre-existing antibody in the blood and also perhaps at mucosal sites is really the first line of defense against infection. Because if the incoming pathogen or the virus is neutralized, or even if the antibody binds to it, is taken up by phagocytic cells, that really, this removed antigen or infection initially is the key mechanism of protective immunity. Of course, more antibody will be made after infection, but it's the pre-existing antibody that's the most critical. I really want to emphasize having pre-existing antibody. And then the second line of defense is the T cell response that kicks in, especially if you have good memory T cells, they will respond very rapidly. The frequency will be higher. Not every incoming pathogen is neutralized by the pre-existing antibody. So some cells will get infected in most cases, but now a rapid T cell response will result in the elimination of those infected cells. It's a simple way of looking at it, but this really is the essential basis of immunological memory. Because also more antibody comes up on recall response from the memory B cells that contributes to the overall control of the infection. So how do we maintain this long-term antibody responses for years after the infection is gone? And as I said, so how does this happen? And this happens primarily because there are a subset of plasma cells that mostly reside in the bone marrow and constitutively secrete antibody for decades, for many years, and very often for decades. So this is a very unique cell, has only one purpose in life, and that is to be constitutively secreting antibody in the absence of any antigenic signal. Plasma cells do not retain the antibody at the cell surface. So there is no mechanism of signaling a plasma cell, fully different plasma cell cannot receive any more signals to the B cell receptor. So the B cell receptor is not on the surface, it is secreted, complete secretion of antibody. Essentially, a plasma cell cannot sense antigen. It's just producing antibody. More than 50% of the protein in these large cells, which have a very large cytoplasm, very large endoplasmic reticulum, very large ER, because they are just pumping out antibody. Essentially, it's a factory for making antibody. These cells, as I said, reside in niches within the bone marrow, and they have the ability to survive long term. They get signals, nurturing signals from stromal cells in the bone marrow that can maintain their life for years, if not decades. They also don't divide. So it's a quiescent cell producing antibody and persisting for many, many years. The first observations of these plasma cells were done in mice by actually work that Mark Slifka, who was a graduate student in my lab, showed in a paper in 1998. And Andreas Radbruch, who's an immunologist in Germany, also has similar observations at the same time. These findings now have been confirmed, extended. Actually, a lot of this work done by Mark Slifka later on in his own lab at Oregon have really confirmed and extended many of these findings in humans. So what about T cell immunity? What does T cell immunity do? Again, very broadly, one can divide this into two subsets. You've got CD4 T cells that do two things. One, they provide help for antibody responses. Without the CD4 T cells, antibody responses are very suboptimal. And without the CD4 T cells, you absolutely get no long-lived plasma cells and no durable antibody responses. So not only are they playing a big role in the initial generation of the antibody response, but also in the generation of these long-lived plasma cells. A key role that the CD4 T cells play is in the formation of the germinal centers. And these CD4 T cells are referred to as T follicular helper cells because they are in the germinal centers talking to these cells, providing the right cues and signals for these B cells to differentiate into long-lived plasma cells. So the germinal center is another key element in the generation of the long-lived plasma cells. And this is again due to CD4 T cell help. But CD4 T cells, in addition to providing help for antibody, can also have antiviral effects. We usually refer to these as Th1 type cells. They make interferon gamma and other cytokines that can have antibodies. CD8 T cells are a little bit different. They are a single-minded weapon. Their main goal is to kill the infected cell. Of course, they do other things. They make cytokines and so on. But these are the professional killers that we have. They are the most efficient cells that our body has for finding, seeking, and then killing the infected cell. They have the ability to home to wherever the viral antigen is, go where the infected cell is, and kill that cell. So the combination of CD4 T cells, CD8 T cells really is critical in terms of help for B cells here for the CD4 and also antiviral effects and then killing of the cells by the CD8 T cells. What about protective immunity, longevity? Again, like the plasma cells, which can live for extended periods, memory T cells can persist long-term in vivo. Unlike the plasma cells, which are constitutively secreting the product antibody, the long-lived memory cells actually are resting memory cells poised to quickly respond. And they undergo a slow homeostatic proliferation, which is antigen independent, usually driven by IL-7 and IL-15. So they have survival, they get survival signals from IL-7 cytokine, some proliferative signals by IL-15. And this is proliferation that just is self-renewal. So they undergo slow self-renewal. A beautiful study actually that Rafi did, was published in Nature 2017, showed that in vivo in humans, these memory cells turn over once every year or once every year and a half. But this allows them to maintain their numbers at reasonably constant levels after they have dropped to a certain memory stage. So again, long-term persistence of memory cells has been shown, and they undergo the slow homeostatic proliferation. The very important property of memory cells is that they can elaborate effector functions almost immediately. So even though they are quiescent, their chromatin is open in the effector stages. Again, this is work that Rafi did in her paper, that they retain those epigenetic marks that happened decades earlier. So that now upon getting TCR signals, immediately interferon gamma is expressed, perforin is expressed, granzyme be expressed, and they can kill the cell. This can happen within hours after re-exposure. A very, very rapid ability to kill infected cells and secrete cytokines. And then this combination of the T and B cells and these properties essentially is what confers protective immunity. And as I mentioned, both CD4 and CD8 T cells can be detected in humans decades after an acute viral infection. And then earlier studies in the mouse model that we use have established these principles. So let me now switch to the data part of my talk, which is on the COVID-19 pandemic. And I don't need to remind any of you of the devastation this pandemic has caused, but as of yesterday or a few days back, estimate that 160 million cases worldwide, 3 million deaths. Almost every country in the world has been impacted by this, and this is by far the largest pandemic of the century. And unfortunately, these numbers are probably a vast underestimate overall, and it's likely that worldwide these numbers are maybe significantly higher, both in terms of the cases and in terms of the deaths. So the first simple question is, why did so many people get infected? And the answer for that actually is very simple, because this is a new virus that emerged, and very few of us were, yeah. What about this? Can you hear me now? Is this better? Hello, is this better?
Yes, better.
Yes, sir.
Sorry, thanks.
Okay, yeah, okay, thank you. Yeah. So the reason so many infections happen so quickly is that the population was immunologically naive. And this is a striking example of how susceptible we can be to a new virus that emerges, and we have no pre-existing immunity to it. It's a terrible way to learn this, but in the past, people have known this. In the more recent, we had forgotten that immunologically naive population, especially if the virus is highly transmissible, highly contagious, and also pathogenic. And unfortunately, all three of these combinations are there with SARS-CoV-2. It is highly transmissible, and in many individuals is really causing very severe disease. And the population is immunologically naive. But one promising thing that appeared very early on when this pandemic started, some very early reports were published from scientists in China, from Wuhan actually, where the first pandemic started, that people who had gotten infected were actually making reasonably good antibody responses quite early. That is, within a month, they could easily detect antibody responses, and some were even, or the neutralization assays at that time had not been optimized, but really binding antibody was encoded. And then a couple of papers came out in the US last year, in the spring last year, and also a paper was published from the Vaccine Central Group. This study was led by Mehul Sutter and Jens Ramat, who are faculty members at the Emory Vaccine Center. And what they reported, and this was actually went on preprint in May 2020, they studied about 45 patients who were at Emory University Hospital. At this time, this was a period, this was March, April of last year, where infection in the US was really very bad. There was a bit of a crisis almost everywhere in the States, especially in New York, with a very large number of deaths. But Atlanta also was getting many infected people. Hospitals were kind of overcrowded. And so Mehul and Jens were able to do antibody analysis in 42 patients that were hospitalized at Emory University Hospital. All of these were fairly severe cases. A couple of these individuals also died. And what was striking to me and to all of us was how quickly the neutralizing antibody responses were detected. Even essentially within 2 weeks, within 2 weeks of confirmed PCR, pretty much everybody had neutralizing antibody. And almost all of them at that time had already converted from IgM to IgG. So there was fairly quick isotype switching and reasonably high frequencies of neutralizing antibody. And what this told us is that this, even though it's a terrible virus, we are naive to it. Actually, this S protein is highly immunogenic, and it's not that difficult to generate neutralizing antibody responses, at least certainly not in the context of a natural infection. Also, work that was done by Jens and also by many other people in the field showed that these early antibodies that came up that were IgG and were able to neutralize SARS-CoV-2, and this was based on making monoclonal antibodies from the patient, is that they were all coming from naive B cells because there was hardly any somatic heart mutation. So this was not some recall response or some cross-reactive response to the common coronavirus, although at a low frequency this could happen. But the majority of the antibodies that were coming up were coming from naive B cells. So to me, this was the first promising sign for that this may be able to tackle this disease because it's not that difficult to make neutralizing antibodies. And this is in very striking contrast, for example, to what one sees with HIV and some other viruses. So it really takes a long time before you get good neutralizing antibodies. Okay, so with that thing, let me get now into our study design. So our study design was to do a longitudinal analysis of virus-specific B and T cell responses in COVID-19 patients. And this is a collaborative study with Julie McElrath at the Fred Hutchinson Cancer Center in Seattle. So we have followed a cohort of about 200 convalescent patients for about 8 months. Actually, now it's longer, but the data I'm showing you is up to 8 months post-confirmed PCR. The cohorts are about equally divided between patients in Atlanta and patients in Seattle. The majority in this study, the majority of the 200 patients that we have analyzed were actually non-hospitalized. There are a few hospitalized patients, about 10% of them were hospitalized. The majority were sick for a few days, some a little bit longer, some a little bit less. All were confirmed PCR, but they were all non-hospitalized. I just point that out, that we're not looking in this case at the very severely ill hospitalized patients. And what did we do in these studies? Oh, just to remind all of you that what the coronavirus looks like. You've all seen this photograph multiple times, but I want to point out to you the spike protein. And some of you may know this, but some of you may not know it, that these group of viruses are called coronaviruses because the term corona means crown, and this looks like a crown. So this is a beautiful protein sticking out, looking like a crown on, and is, as I mentioned to you, also highly immunogenic. It's actually a very large glycoprotein, about over 1,200 amino acids, which is quite unusual for a viral glycoprotein, usually much smaller than that, but really sticks out. And in this case, as you all know, binds to the ACE2 receptor. This is a little bit more of a cartoon structure. I want to illustrate here one main point. As I mentioned to you, the whole spike protein is about 1,200 amino acids in length. The receptor, 1273. The receptor binding domain, this is the region of the spike protein which binds to the ACE2 receptor. This is about 200 amino acids, and really this is the part of the spike protein that binds to the ACE2 entry receptor. So in our study, we did the following measurements. So first, we looked at binding antibody to SARS-CoV-2 spike protein in all of these 200 plus individuals. And one thing I want to stress is this truly is a longitudinal study, because for all of these over 200 patients, we've got between 2 time points and in some cases up to 7 time points in this 8-month window. And over 60% of our patients that we followed are between 3 time points, between 4 time points and 7 time points. I emphasize this because there's quite a bit of variation in the level of antibody between different individuals. So cross-sectional studies, which are very useful, can be a little bit misleading because you don't know which dot joins to the next dot in those things. You just take an average and you make a conclusion. Again, those questions are not wrong, but the elegance of longitudinal, where you can truly draw and connect lines, is really adds a bit of clarity and elegance to the study. So this truly is a longitudinal study looking. So we looked at binding antibody to the SARS spike protein. And then I will show you what is the effect of this SARS-CoV-2 infection on antibody titers to the common alpha and beta coronaviruses. And there are some interesting observations that we made here. I will then show you data on neutralizing antibody titers to SARS-CoV-2. And this is using a live neutralization assay. And I emphasize this because the pseudovirus neutralization assays are useful, but there are some caveats with that. So the live neutralization assay is always the better one. And Mehul Sutter at the Vaccine Center has played the lead role in doing all the neutralization assays. And of course, this has to be done in a DSMP containment facility. We then looked at memory B cell responses to SARS-CoV-2, and then looked at CD4 and CD8 T cell responses. So it's a very comprehensive look at not only binding antibody, but also neutralizing antibody, and then looking at memory B cells, memory CD4 cells, and memory CD8 T cells. So I'll start with the binding antibody data. It's a busy slide, but I'll walk you through it. And essentially what we did was we looked at binding antibody to the full length spike, to the RBD region, and to this N-terminal domain region. The findings are very similar for all three, so I will focus only on the spike protein. And I quickly point out what we see with the others. We've looked at IgG, we've looked at IgA, and also at IgM. But let me focus on the IgG response because this is the one that's most durable and of the highest titers. So we looked at 222 COVID-19 convalescent patients, and we looked at days post-symptom onset / confirmed PCR. And we also had samples from 51 individuals pre-pandemic. And actually, these data are from cohorts from 2013, 14, and 2017, 18. So we were doing studies either of people getting the influenza vaccine or people getting the yellow fever vaccine, studies that actually Rama was very involved in. And this allowed us to do longitudinal analysis of pre-pandemic healthy individuals. Essentially, what this shows is that there really isn't any binding antibody, either IgG to the spike, to the RBD, and the NTD actually is also very low. And so essentially, this is our negative control. And what you see over here with the COVID-19 patients, and this is a log scale, really a massive log2 actually is increasing, really a massive increase in IgG binding antibody to spike protein to the RBD, less to the NTD, but very high to spike and RBD. And then we asked the question, what is the half-life of this of these antibody responses? There are two ways of measuring the half-life. One is do exponential decay, And this gives the exponential decay rate of about 120 days for this, again 116, about 130. But it is a second way of doing this, which is using a method called the power law method. And this allows you to look at if there is a biphasic aspect to the decay. That is a little bit faster decay earlier, and then a little bit of a slower decay. In fact, you can already even see that by looking at these, some of these a little bit faster decay and then a slower decay. So this power law method calculates the biphasic aspects of this curve. And using the power law method, what we find is that starting around day 120, there is actually a little bit of a biphasic aspect. And what this half-life shows you is that if from day 120 onwards, you could even do that a little bit earlier, but from day 120 on, day or 100 or 80 onwards, you now have a slower half-life decay. So this comes out to about 238 days for the spike protein, and again in the 200-day range for the other ones. The IgG also comes up and then also has a reasonably good half-life. IgM comes up and decays as expected, but faster. So the power law is method showing us a biphasic aspect is actually very encouraging in terms of the longevity of the responses. As I mentioned to you earlier, the long-term antibody responses are really maintained by these long-lived plasma cells that reside in the bone marrow. Whereas the early response comes predominantly from plasmablasts that have been recently generated, in this case from naive B cells that have proliferated and differentiated into antibody-secreting cells. Majority of these actually are short-lived. They need to be because you just make it too many plasmablasts to retain all of them. Our body would get clogged up with too many plasmablasts if all of them survived. So this first response is by the shorter-lived plasmablasts, and as these are decaying, we're now getting some, at least, longer-lived plasma cells. It's a very early, encouraging sign about the long-term longevity of the immune response, of the antibody response. We then asked the question about what is happening to the common alpha and beta coronavirus antibodies in these COVID-19 patients. And just to give you a little bit of a background on the common alpha and beta coronaviruses, there are two alpha coronaviruses, 229E and NL63, and two beta common coronaviruses, HKU1 and OC43. These are what we refer to as the common coronaviruses. They usually, these infections are usually acquired during childhood. And by the time one is an adult, there is very high seropositivity. Certainly in the US, over 90% of the population is seropositive. I presume it would be the same in other parts of the world. They are found worldwide. They do cause some cold-like symptoms, but none of them are that severe. But there is high seropositivity. And SARS-CoV-2 is a beta coronavirus. So there's some homology with these. And I'll show you now two data slides. One will be, if you just look at the antibody to the common coronaviruses in the pre-pandemic sera, what does one get? And then I'll show you what is happening in the people who have gotten infected. I will also show you data on SARS-CoV-1, which is a beta coronavirus, but of course, exposure of this was very limited to the few people who were infected with SARS-CoV-1 when this virus emerged about a decade ago. Okay. So let's look now in the pre-pandemic sera and see what do antibody responses look like to the alpha and the beta coronavirus. So these are that same 51 patients from that I showed you earlier in the red lines from 2013-14. Of course, all of them turned out to be seropositive to the alpha and beta common coronaviruses. And here are the longitudinal analysis of these pre-pandemic sera. So here is IgG antibody to 229E, to NL63, and here is to HKU1 and OC43. And of course, none of these people had been exposed to SARS-CoV-1, so it was negative. So what strikes you from this thing? What is the most striking thing? One of the most striking thing here is they're straight lines. It was like as if I took a ruler, or Susie, who did all this work, took a ruler and drew some straight line. there's absolutely no decline in this 200-day window that we have followed these individuals from several years back. And I think if we do more, we will see the same thing. Okay? So what this tells us, again, it goes back to how this antibody being maintained. It's the long-lived plasma cells. So these people were exposed as children. Antibodies was, I'm sure, higher at one point and has come down. And now this is a plateau that's maintaining the plateau antibody and maintained by their long-lived plasma cells. There's a lot of misinformation in the literature about immunity and antibody responses to the common coronaviruses decaying, and the data is just wrong in terms of antibody. Can these people get reinfected with coronaviruses to some degree? Probably yes, because sterilizing immunity is a bit of a myth. Some re-exposure can certainly happen, but doesn't look like it happened in any of these individuals in this window because we see no spikes at all in the antibody. Okay, so now we ask the question: what happens when you get infection with COVID-19? What happens to these antibody levels? The data actually is quite interesting. Here now, let's look at the blue lines are the COVID-19 patients that I showed you data on earlier, 222 of them, and pre-pandemic 50. You can see that for the alpha coronaviruses, you again get now, you had 51 straight lines before, you've now got an additional 252 straight lines over here for 229E and for NL63, which shows that again, in these individuals, there was long-term maintenance of this, very stable maintenance of these alpha coronavirus IgG antibodies, and there was really no effect of COVID-19 infection on antibody levels to the alpha coronavirus. Not surprising because there's less homology. Interesting pattern with the two beta coronaviruses. Perhaps a slight increase in this. What that means is some of the antibodies induced by the SARS-CoV-2 infection might be cross-reacting with HKU1. If we had just looked at the blue lines, we would not have made this conclusion, but because we have the red lines slightly higher. And these antibodies do have a decay because this is induced by SARS-CoV-2 infection, early plasmablast response, and cross-reacting with HKU1. More convincing with OC43, because you can see the blue lines are more higher over here compared to the red. Again, a decline in these. The old antibodies are from the long-lived plasma cells, new antibodies generated by the plasmablast with a decay of cross-reacting with OC43. And then here's data with SARS-CoV-1. Clearly, because the baseline was very low, clearly an increase. So almost everyone who got SARS-CoV-2 infection in our study is making some cross-reactive antibody to SARS-CoV-1. We are now doing neutralization assays on neutralization to see if any of this is neutralizing and might be conferring some protection to SARS-CoV-1, even though there's no evidence it's around. But could be of some interest in terms of people talking about a pan-coronavirus vaccine, is that this one is inducing antibody to SARS-CoV-1 also. Okay, now let me switch to looking at neutralizing antibody. So here is the neutralizing antibody data. There's a little bit more play in this because the scale is such that you can get more variation. But the bottom line here is that the vast majority of individuals, in this case, N is 183, that the majority of individuals who were infected in our study that we followed still have neutralizing antibody responses. Again, the half-life by the power law is about 250 days, and the exponential decay is 150 days. We then ask the question, what is the correlation between neutralizing antibody and binding antibody? And as others have reported in multiple papers, and also this was reported in the original paper by Nehul and Jens, there's a pretty good correlation. So here's the correlation between the neutralizing antibody to the spike, to the neutralizing antibody to the virus and binding antibody to spike, and reasonably good correlation. And this is the RBD, and again, a very good correlation. Okay, next question we asked was, what about memory B cell responses? Interesting kinetics for the memory B cells. So memory B cell responses actually were very surprisingly high. And we had up to, on an average, about half a percent of the total IgG memory B cells that we have, or these people had, were actually specific for SARS-CoV-2 spike protein. This is a high frequency after a single infection. And not only was a very robust memory B cell response induced, but also these numbers continued to actually increase up to 70 to 80 days, and then started plateauing, but there was really no drop seen. So very interesting pattern. Nice increase in memory B cell numbers, coming up to 0.5%, then kind of being maintained at that, numbers increasing for longer period than expected. And I think part of this may relate to redistribution of more cells coming from germinal centers. I think we're getting a lot of memory B cells formed in germinal centers within the first few months. GCs can persist for several weeks, if not months. And so the bottom line here, robust memory B cell responses clearly persisting without showing any appreciable decline over this 250 window. We then asked how many of the memory B cells are to the RBD region and how many are to the rest of the spike. And the ratio is very similar to what the size is. So we found that, this data is shown on the next slide, we found that about 25% of the memory B cells that bind the spike protein are actually specific for RBD, and that's shown over here. And the remaining 75% are to other regions of the spike protein. So I would emphasize an important aspect of protective immunity here. So the key antibody that neutralizes the virus binds to the RBD. But I think antibody to other parts of the spike protein will also have potential to be protective in vivo, because it can result in FC-mediated effector functions, and in particular, in the phagocytosis and removal of the viral particle. I think virologists tend to overstress the neutralizing assay, because indeed it is a fantastic assay and is probably the most efficient way of neutralizing the infection. But binding antibody and removal of the viral particles, which is coated by antibody by phagocytic mechanism, is a very important mechanism of protection and should never be underestimated in terms of its role. And I point this out because there is always concern with the variants coming up.
Yeah, I'm not sure I can do that. This is Chandler.
Hello.
Okay. Maybe you can start with.
Can everyone please shut their video audio off?
There's a part of me that always knew I wanted more. All right,
No.
there's a nuclear holocaust.
Tahid, you can switch off that
Can you please, can others mute themselves, please? Okay, good. All right. So, so I was just pointing out the importance of binding antibody to non-RBD regions also playing a role in protection. Okay, now let me switch to the CD4 and CD8 T cell responses. So in the earlier data I showed to you for the antibody and for the memory B cell, the focus was on the spike protein and looking at the RBD region. Here, the CD4 and CD8 T cell assays were done to look at CD4, CD8 responses, not just in the spike region, but also in the nucleocapsid protein, in the envelope protein, in the matrix protein, and some of the other open reading things. Not the entire genome, but several other regions in addition to the S protein. And here the data is not in 222 patients at this stage, but is like 114. And here is a summary of CD4 T cells that either made interferon gamma or IL-2 or CD40 ligand, basically a composite of the total CD4 response to SARS-CoV-2 peptides that we do, which is spanning maybe about 25 to 30% of the genome. Okay? And again, pretty good frequency, about 0.5 to 1% of the CD4 T cells are specific for SARS-CoV-2. Very nice studies done by Shane Crotty and Alex Seke have also made similar observations in the convalescent patients. that you can get CD4 frequencies in this range. And when we do the half-life, it's about 207 days for CD4 cells. And if you ask the question, what is the phenotype of these cells based on this canonical central and effector memory definitions, and we find that the majority of these cells have a central memory phenotype, and some of them have this effector memory, and the stemra population is not present in the CD4. So this is also a good result. What it shows us is that these central memory populations, which are basically circulating in blood, also go to other lymphoid tissues, usually have a longer half-life. And again, you can see that their decay is actually less than the decay of this more effector memory population. As again, the very preliminary at this stage, but suggestive of nice long-lived central memory cells that can look at. And they're also very much of a Th1 type. So if you look at the functionality and of these cells, they're polyfunctional. This is a very busy slide, but the bottom line is that if you look at what these cells do functionally, many of them are multifunctional, polyfunctional, making from gamma, IL-2, make TNF-alpha, CD40 ligand. There also will be TFH-like cells, which I'm not showing you here. There'll be more within germ centers. You can also see a few with TFH properties in the blood. But essentially, the bottom line is you're getting a durable CD4 response that's fairly polyfunctional. And this is data at the day 30 for functionality. And if you look at the later time point again, you see that same polyfunctional cells are persisting. What about CD8 T cells? So here's the CD8 T cell response. And again, similar analysis was done using these overlapping peptides. Frequency of CD8 actually on an average is slightly lower than CD4s, but 0.2% of the CD8 T cells, again, half-life of about 200 days. And then we looked again at that canonical way of looking at the different subsets. And the results here actually are quite interesting. And what we find is that that the effector memory population, that is, would be the more recently generated effectors, that these are declining as one would expect because they're changing the phenotype and some of them are dying. But this stemra population, and that Rama defined in her yellow fever studies, actually is increasing. And these are the cells that give rise to the very long-lived memory T cells that one can find decades later. So again, here there's an indication of the generation of these long-lived T cells. Like we are seeing a hint of the generation of the long-lived plasma cells, we're also seeing hints here within this first 8 months to a year of the generation of CD8 T cells and CD4 T cells that could persist long-term. And again, polyfunctionality-wise, these cells are highly polyfunctional, make multiple cytokines at the early phase and also at the later time. This is an important point. So don't look at the slide here before I set it up. So what this shows is the breakdown of what are the proteins or open reading frames of SARS-CoV-2 that are recognized by CD4 T cells and by CD8 T cells. So this is a breakdown of how the CD4 responses are distributed. That is, you have many targeting the S. This S1, S2 is just how the peptides were made. So this is simply one part of the S spike protein, another part of spike protein. Basically, you see that there's good responses to different parts of the spike protein. There's good responses to the envelope and the matrix in the peptide pools corresponding to these two. Good response to the nucleic acid and to two of these open reading frames. So this tells you there's a very broad response, recognizing many different regions, many peptides coming from many different parts of the SARS-CoV-2 genome, which is good. But we see a very different and interesting pattern with the CD8 responses. Of course, there are CD8 epitopes. T cells seeing CD8 CD8 is seeing epitopes in the spike protein, in the E and M, and in these ORF, but the majority of the responses are directed to the nucleic acid protein. And this is a very important point because what this tells us is that when you have a SARS-CoV-2 infected cell, the most dominant epitopes that are displayed on the infected cell are coming from the nucleic acid protein. It's not that theoretically there are no epitopes in the S, but the mechanism of antigen processing, the abundance of the protein, that very much dictates what peptides are put on the infected cell. In this case, the nucleic acid protein is displaying the largest number of class 1 restricted epitopes on its infected cell. And that's the reason why the CD8 responses to that protein are much higher. And this has a lot of implications in terms of vaccine design. Currently, none of the licensed vaccines have the N protein. It's all S protein only. And those vaccines that can induce CD8 responses, like the RNA vaccines and the ad vector vaccines, the protein plus adjuvant or inactivated don't do this well, but certainly the ad vectors induce good CD8. mRNA vaccines potentially can induce very good CD8 responses. So it would be good to include the nucleic acid protein as we move forward, because this will be very broad and good protection. So I would emphasize the importance of including the nucleic acid protein in SARS-CoV-2 vaccines for inducing and providing the most optimal coverage and breadth of the CD8 T cell response. Okay, let me now finish this part by making some concluding comments. So what we have shown is that there is good durability, for at least for over 8 months. Circulating IgG antibodies present with a power law decay of about 230 days. Neutralizing antibody also power law decay, suggesting that there is going to be longevity in these responses. Memory B cells, we have interestingly no decline. And at what point numbers will come down and where they will stabilize will be interesting to look at. But this is a very good sign for the moment, because it means that upon reinfection, there will be a rapid recall response, not only the pre-existing antibody, but there should be a rapid recall response coming from the memory B cells. Again, polyfunctional CD4 and CD8 T cells showing the right indication for longevity in humans. So the conclusion is very broad and balanced immune memory. Then we get to the issue of will this give protective immunity? My prediction at this stage would be yes. This is not only high frequencies and high levels, but it's also very broad. You've got binding antibody, you've got neutralizing antibody, you've got high level of memory B cells, and you've got good CD4 and CD8 T cells. It may not necessarily be sterilizing immunity, but if these people get infected, they will probably control the infection much faster and will not have severe disease. I think I would be very surprised that if a large number of reinfected, of course, there are always outliers. Some people do have low levels, so there will be some breakthrough, but majority of these individuals should be protected from severe disease for sure. Okay. But what about the variants? This brings in some interesting caveats and interesting questions that we don't fully have the answers for. Okay. But I want to address this in the context of my next question. And the next question is, what will protective immunity look like that is induced by the SARS-CoV-2 natural infection versus the SARS-CoV-2 vaccine? And there are many differences between natural immunity by the natural infection versus immunity by the SARS-CoV-2 vaccine. We fortunately have very good vaccines, and even the vaccines which are not that as good as the other ones do confer protection. So the good thing is that almost every vaccine that has been made has some effect, and which is fantastic, because what it reinforces is any vaccine is better than not getting a vaccine, because the person who's most susceptible is the naive individual. So vaccination is really highly recommended, no matter whether it's the best vaccine or the second best or the third best vaccine. Vaccination should be given to everyone, and the best available vaccine should be used. But what will be immunity look like, longevity look like? I think these are unknown questions at the moment. The data coming out from some of the vaccines looking at antibody responses, durability seems okay, but again, we don't have the kind of detailed data that I just showed you with the infected people. But the bigger difference is that here we are relying on just the S protein. And in some cases, we will not be getting very good CD8 responses, especially with the inactivated and the protein-only vaccines that will be antibody and CD4. This one is going to give you very broad protection. So if one is confronted with variants, the acutely infected people who have survived are likely to probably do better because they will have other mechanisms. So one of the things we need to do, I think, in the future as we move forward is really include some other proteins into the SARS-CoV-2 vaccine. In particular, I would highly recommend including the nucleic acid protein with vectors that induce CD8 response. And again, this equally big question over here is how will different immunity long-term by different SARS-CoV-2 vaccines? Again, these are data that will come and we will learn from these. But I would say learning lessons from what we're seeing in the natural infection should be applied as we try and start addressing issues of longevity of the responses. Right now, we are at the early stage, vaccines are working. How will it be 2 years from now or 5 years from now? I think there a lot of thought needs to be given, and we need to be learning what we learned from the natural infection. Okay, again, as I mentioned to you earlier, this was a collaboration between the Fred Hutchinson group, Judy McElrath's group, wonderful scientists over there. Our group, I want to acknowledge in particular Susie Linderman from my lab, who was the lead person doing all the serological analysis. Many other people from the lab contributed. Jens Rammert lab was involved. Sutter's lab did all the utilization assays. I have to acknowledge very much our clinical team that recruited all of the patients in Emory, and similarly Julie's team that recruited the patients in Seattle. Our modelers who helped us with the power law analysis, funding from the NIH and funding from the Donald Trust, with generous support from this Trust Foundation, and most importantly, really the patients who agreed to this and are really cooperating and have also agreed now to be in the study for 2 years or even longer. And then some of these patients are now getting the vaccines. That gives us an opportunity to see how they make recall responses upon vaccination. Just if you bear with me for 2 minutes, I want to switch a little bit here and just tell you in less than 3 minutes what our lab at ICGB is doing. So this was a ICGB Emory Lung partnership that started many years back, and this is the opening ceremony. This is the dean from our medical school, associate dean, and this is Kapil Sibal, who was actually the science minister at that time, and they came to the ceremony. We've been very fortunate. We have a wonderful group of about 15 people who work closely in collaboration with other scientists and clinicians in India. I should also acknowledge that Shahid played a major role in establishing this relationship. He was, as we all know, at the ICGP at that time. So what has our group been doing for all these years? Actually, our focus primarily has been on human immunology and human viral infections. And a major focus of the lab has been on dengue virus infection in India. We've published several papers, a lot of interesting ongoing studies. But then in the last year, the group pivoted very strongly to doing COVID work. And in collaboration with DBT and ICMR, our group was able to get samples from many COVID-19 patients in India. They've characterized their immune responses, but most importantly, they have generated a very large panel of human monoclonal antibodies against SARS-CoV-2. And this was done by using the RBD protein to sort RBD-specific B cells from these COVID-19 patients in India, clone out the heavy and light chain, and then express it and make monoclonal. This is a technique that we developed many years ago, which is a very rapid method of making human monoclonal antibodies from the patient themselves using either antigen-based or directly from the plasma. In this case, these were made using the RB2 antigen. And early data shows that several of these actually are very good neutralizers. Currently, studies are ongoing to see if these monoclonals will also neutralize the variants that are currently circulating in India. So we will keep you posted and are very excited and have given this a very high priority. The Indian government, ICMR, DBT, and ICGB are in the process of patenting these monoclonal antibodies. So they potentially could be used for therapeutics and also in some cases for prophylactic if these turned out to be this is our group. This is an earlier picture, but I want to point out to you two people who have played a lead role in running our ICGP lab. One is Anmol Chandali, who's based at ICGP in India. 100% of the time she's a fantastic scientist and a leader, providing training and oversight of many of the postdocs and graduate students that we have. And Murali Krishna, who is a faculty member at Emory and goes back and forth between India, between the ICGP lab in India and Emory University. And of course, we will recognize Shahid over here, who's played a very big role in nurturing our center. Thank you very much, and happy to answer any questions.
Thank you very much, Rafi, for a very comprehensive talk. Let me explain the ground rules here for people who want to ask questions. Many of you have already put questions in the chat box, and I will go down through them in the order in which they appeared. Please allow me to use my prerogative as a host to ask you the first question. The most common question that we get here is, of the two vaccines in India, which one is better? Covaxin, which is a whole virus vaccine, and or the viral vector spike protein Covishield. I do understand that whole virus will, has many more proteins, but my specific question is, would killed viruses really generate good CD8 cells? And so what's your take on it?
Oh yeah, I mean, that data exists. It's not my take, and it's not a specific comment on the Covaxin, and it's a general comment on inactivated viral vaccines. They don't generate CD8 response because it's essentially, but they're quite efficient in generating antibody because they're seeing the whole particle, and they will generate good CD4 responses. And many of our most successful vaccines historically have been inactivated vaccines. So the platform is very good because it does induce good CD4 responses and can actually be very immunogenic in terms of antibody because you are seeing the whole particle there. So inactivated vaccines is a good platform, but as far as CD8s are concerned, no, you don't get that with inactivated vaccines.
Right. Okay, so I think the Dr. Sohaib Khan has a hand up. Do you have a question to ask? If you have a question, please unmute yourself and ask. Or maybe the hand went up by mistake. Okay. Can I invite Mehak Vora to unmute herself and ask a question? She was the first one to put something in the chat box.
Hello, good evening, sir. It was a brilliant talk. I just have one question. You have shown in your data that there does occur the memory response for a longer period of time. So why do we currently see in the current situation people getting reinfected even within a month from recovery? So what's your take on it?
Yeah, I know. I mean, that's a very interesting point. I'm glad you raised that. And I mean, I don't know the situation in India, what is happening in terms of what was the level of antibody responses or T cell responses in the infected. And I think this is something that needs to be done in more detail. But based on the US data, the number of people who recovered from an acute infection, really there are hardly any reports of those people being reinfected. It's not that it cannot happen, but it's very, very rare, if any. Now, so I'm talking there about the infected people. In terms of vaccines, I think this is more of an open question because we don't know how immunogenic that vaccine was in that individual. We don't know how efficient those antibodies were in terms of neutralization of the variants which are circulating. So I think breakthrough with vaccine could be a bigger problem. And that was what I was pointing out earlier, is that we are relying predominantly on the S protein only. And there could be more breakthroughs if there's a variant that especially is mutated many of the neutralizing epitopes and is more transmissible and more infectious. So I think there could be breakthroughs there. But one needs to kind of get a little bit more robust data from India in terms of how many infected people really are getting reinfected. That has not been reported, certainly in the US. If it is, it's very rare, if at all. And I think also from Europe, it's very rare. So one needs to look at more detail at what's happening here.
Thank you, Rafi. Khushbu Singhal, could you unmute yourself and ask? And may I request people to keep their questions really short, because we have many people who want to ask questions.
Yeah, I'll keep my answer short too.
Hello, sir. That was a very informative talk, and I just wanted to ask what extra benefit or what extra immunity will vaccines provide if to someone who already has been infected with SARS-CoV-2? Because if the person is
yeah, that's also a very interesting question. And I think getting a vaccine is probably good, because I think in the majority of the individuals, probably the vaccine is not needed. But as you even saw in the data that I showed you, there are some people who recovered from the acute infection and the responses are very low. And this usually happens in people who had a very mild infection. So the antibody response, we don't have very good data on the T cells on that, but the antibody response does correlate with severity of disease. So the people who were most severely infected have higher levels of antibody than people who had a very mild infection. Their responses are lower. And since we are dealing with a very large number of people, I think there will be individuals who were acutely infected, but if the infection was very low, their antibody and T cells might be very low. So I think for those individuals, getting a booster shot is good. So I would say that if the vaccine is available, you should get the vaccine. People who have been infected and recovered should also get the vaccine. There's certainly no harm, and it would, even if you're high, it will make it higher. If you're low, it would give it a better boost. Memory B cell numbers, I told you, in our case, even in those who were very low for other things, still they were there. Yeah. So I think the memory B cell numbers are very promising. So there will be good recall responses from all of these convalescent people. In fact, that's being seen now when they're getting vaccinated or vaccinated. Very good recall response.
Thank you. Nawaz Akhtar, unmute yourself and ask.
Yes, sir. Sir, it was a very amazing lecture. Thank you so much, sir. Sir, my question is, what if any patient take 2 doses of Covaxin, then after he takes another live attenuated vaccine?
Oh, you're saying it's some kind of a mix and match?
No, I think he's saying take both doses of one vaccine and they take another booster of a different vaccine.
Yeah, I think this is one of the questions that everyone asks. And my answer to that is yes, I think it will help, but we have no clinical trial data. So the vaccines follow certain rules. The people who make the vaccine spend a lot of time and money doing a controlled trial. And the usual practice of vaccination is that a vaccine has been approved based on that clinical data, which is very robust and takes time and huge amount of money and effort. And so one usually sticks to that regimen. One doesn't start. But now we have a situation where there are many vaccines. Usually we don't have that many vaccines for a given disease. Now we've got probably 10 at least, maybe even more. And that number is growing. So there's a lot of discussion. Can I just take this and add that? And my answer to that is, certainly there's no data, clinical data, what it does. Common sense says that, okay, if you take another one, it should increase antibody response and it should be okay. But I think we'd be breaking from the practice of how vaccinations are done. And the concern with that is sometimes there are adverse events, but they wouldn't have been tested. So I think there's a risk here and also a bit of a danger in terms of bad press for vaccination. If people decide on their own to take different vaccines, combine them, there's no clinical data that exists on it in terms of benefit or in terms of concern. Now, I have always discouraged that cowboy type of approach, saying, okay, let's do this. I think one should follow the rules of vaccination. I was also opposed to arbitrarily increasing the interval between the first and the second, because the trials really were not done for them. And if you give one shot and then leave a long period of interval, the protective immunity after the first shot is really not optimal. So you increase the window of vulnerability. So I'm putting on the hat of of a vaccinologist more from an administrative point of view and saying, one should really follow rules that were defined by the rigorous vaccine trials that have been.
Rafi, interesting you said that, because one of the things that is being hotly debated in India right now is what should be the duration between 2 doses of Covishield. Should it be 4 weeks, which was tested, or should it be 8 weeks, or should it be 12 weeks, or now it has gone up to 16 weeks? And it's all coming from that Lancet meta-analysis.
Yeah. All I'm telling you is that, yes, scientifically, a longer duration is better. In fact, I'm going back against my own science, because we were the first ones to tell people, don't give your vaccines too quickly. Give it time. The period of rest is good for memory T cell formation. This was a paper we published in early 2000s, Sutek's paper, showing that the period of rest is needed to get optimal boosting from the memory cells. But so I'm putting here the science aside a little bit. I never do that, but in this case, I'm putting it aside, even my own science, okay? Because I'm going by vaccine trial. It's not that easy. And you know how bad press can happen. Moderna and Pfizer and AstraZeneca did study with a given interval. And whatever data they produced is based on that. Now, if you arbitrarily increase it, as I said, the window of vulnerability is more. And if you give it later, scientifically, I don't think it'll be any worse. Might even be better. But what about the window after the first shot that increases? And certainly you do need that second shot to get the protective level. So my concern here in the setting of the pandemic is that one should be careful that the window of vulnerability not be too much.
Yeah, unfortunately in India, we no longer have that choice.
Yes, but
yeah.
I would say just get the full regimen and I think you'll be okay.
Yeah. All right. Sheetal, please unmute yourself and ask. Sheetal, are you there? Unmute.
Yeah, hi. Hi, Dr. Rafi. Thanks for an excellent talk. So my question was related to the CD4 T cell responses that you showed. And I remember the range for the maximum response was something like 0.1% to 2% of T cells activating in response to the antigen. So is it known for any viral infection, what is the optimal percentage of CD4 T cells such that you have a protective humoral response upon a second exposure?
Yeah, very good question. There are no hard numbers in terms of what the protective threshold is. Nobody knows that for any human infection. But the frequency of but CD4 is actually a little bit higher. The CD8s were the numbers you're mentioning. But again, you're in the right ballpark. CD4 is about 0.5%. To me, that's a lot of cells, because if you convert that into absolute numbers, we have about 10 to the 12 CD4 T cells in an adult human. So that's a lot of cells. That's several billion CD4 T cells. So I was surprised that the response actually was so good after a single infection. So right now I feel good about the frequency of the CD4 T cells, and also the CD8 T cells, a little bit lower, but again, not bad. Where they will plateau, because there is this decline, and what they will be 2 years from now or 5 years from now, that we don't know. That's the more interesting question. But again, hints from some of our studies are that there should be at least a subset of those that persist long term.
Thank you. Morifat Ahmed, unmute and ask.
Yeah, hello, sir. My question is that what will be the effect of these binding antibodies? So if there is mutation in the spike protein, so do these binding antibodies have any kind of efficacy against these viruses, or the virus will render unable to enter into the cell?
Yeah, good question. So the antibodies that bind to the RBD region, there are 2 or 3 key contact regions with ACE2 receptor. Those are the only ones that would prevent actually binding to the ACE2 receptor. So if those regions are heavily mutated, then the ability to neutralize, that is prevent binding to the ACE2 receptor, will be less efficient. And that is a concern, especially with some of the variants which are showing more mutations in that region. In terms of the ability of binding and mediating, so in that case, you'd also lose binding to that region. So you lose neutralization and binding, both will be lost. But where there aren't mutations to the other regions of the spike protein, those antibodies will not prevent binding to ACE2 receptor, but they could be removed from the system by phagocytosis and Fc-mediated effector function. So I think those antibodies, as has been shown in many other infections and models, animal models, those antibodies will also have a role in protective immunity.
Thank you. Vasistha Adiga, please
Hi.
ask your question.
Yeah. So does pre-COVID sample has been tested for SARS-CoV-2 peptide for the T cell response? And if yes, does it show CD4 and CD8 response to SARS-CoV-2 peptides in S1 and S2?
Yeah, it's a very good question. So most of the T cell data I showed to you was actually done at the Fred Hutchinson lab. We're also doing it, but the data that I showed you, and they have all, I didn't show you that, they've also done extensive epitope analysis with pre-pandemic setup. They usually see nothing to the spike protein at all, either very few, if any, to the CD4 or CD8, very low that they pick up in their assay. But to the nucleic acid, they pick up occasionally CD4 and CD8 response. So I think that there's more cross-reactivity.
Is that the reason why we see CD8 response very high for N peptide?
Yes, it could be. It could be that the CD8 response is, part of it is novel epitopes, and part of it could be cross-reactive. Yes. Again, goes back to the importance of the nucleic acid protein in these coronavirus infections.
Yeah, thank you.
Anna Vyakarna.
Yeah, hi. That was a super talk. I'm just trying to think over here that the vast majority of the individuals that you've studied are subjects who are not hospitalized, if I've got that right.
Yes.
So how does this immune profile compare with those with severe disease that has implications in terms of durability, that has implications for both vaccine thinking and reinfection?
Yeah, excellent question. I think some people have published data. In our case, only about 10% of the people had severe disease and they're hospitalized. So, and interestingly, in those, but N is very small. It was only like about 10 that we had. So I wouldn't overconclude from an N of 10. Their antibody responses and T cell response actually were higher in magnitude.
Mm-hmm.
The durability slope was similar, but the magnitude was higher.
Mm-hmm.
And the earlier paper I flashed on one of my slides, but didn't show any data from, those were all hospitalized patients. And we examined the first 2 months. Their antibody titers actually were higher.
Mm.
And it seems counterintuitive, but actually from an immunological point of view, it makes sense, because the more the antigen, the higher the immune response. And that's the reason for it.
Mm.
But would be good. I'm sure that we don't have that cohort, but I'm sure that someone might have it. So you might see more longevity data with the more severely.
Yeah, the question of durability in those subjects and how that then compares with the asymptomatics and the vaccine responses. Absolutely.
I think again in the asymptomatic, the titers, but the really interesting question would be what you just said, longevity with the vaccine responses, because they may not be exactly the same as the infection people, and different vaccines may have different durability.
Absolutely.
To me, that's the other very open question.
Thank you. Yeah.
Jacob John, please go ahead. Jacob John, are you there? Okay, maybe we can move on. Parul Mehrotra.
Hi, thank you for the very reassuring talk, and it was very helpful. Probably a lot of us will sleep better tonight. So my question is to the use of glucocorticoids in the treatment regime. Have you or do you have any indication of how glucocorticoid treatment that is actually in India? I know a lot of people who are being put on glucocorticoids on the day one of getting fever. So how this will interact with memory development, because there's a lot of conflicting evidence of how corticosteroids interact with T cell development and memory.
Yeah, this is a very, very important question. And we have not addressed it because in our case, hardly any of them were on the table. That is an important question. I don't have an answer. And I think one needs to look at it because steroids can be immunosuppressive. My one thing that's a little bit reassuring to me is, although this is pure speculation, is that the steroids are probably being used after the immune response has been generated. It was not that steroids, unless people are taking steroids even before they have any symptoms, the effect should not be that great. But where we will get some data on this is there are currently studies going on in many places, including at Emory, in populations which are either autoimmune, because they are on different kind of immunosuppressive drugs. And our lab is involved in looking at immune responses in lung cancer patients. So I think data that will come from many groups looking at these specialized populations might start giving some indication of what the different treatments might be.
Okay, thank you so much.
Imtiaz Ahmed, you have your hand up. Please go ahead. Okay. Kasturi Pal.
Thank you so much for the very insightful talk. So my question was kind of brushed upon by Dr. Jamil, who took the prerogative as the first question asker. So I was asking whether the different vaccines like mRNA vaccines versus the adenoviral delivery mechanism or live attenuated, which of these would be giving more stronger or longer immune memory? But I guess you kind of answered that. So
yeah, I mean, I can tell you just from the published data what has been. We don't have any first-hand data in our lab, but a lot of very nice studies have been published. The durability part is still open. I think that you will probably get more data on it from the group for studying this. But in terms of immune responses, and this is an objective analysis of what has been published, is if you look at antibody responses, the RNA vaccines do give very good antibody responses, both in terms of binding and neutralization. Both Moderna and the Pfizer vaccine are really excellent induction of antibody responses. If you look at the ad platforms, and this is data from AstraZeneca and data from Johnson & Johnson, and there you get antibody response. It's not as high as in terms of neutralization, not as high as the two RNA vaccines. Novavax, which is not licensed yet, did give very good antibody responses. Novavax is protein plus adjuvant. So the neutralizing antibody responses have been good with the with the RNA vaccines, with the protein, but at least with the protein plus adjuvant. The inactivated vaccine data, I mean, I haven't seen it in detail, but in principle, that platform should give good neutralizing antibody responses, especially if it's combined with a good vaccine, with a good adjuvant. But in terms of the T cell responses, the CD4, I think all of these platforms should give you good CD4 responses, and they do. Again, the Moderna, Pfizer seem slightly better in that. The CD8 responses, inactivated vaccines will be very poor, if any. And in terms of CD8, the ad vector should be good. Although the data that has been published has not been that extensive on the CD8, but the platform based on work that's been done extensively with HIV vaccines or malaria vaccines and TB vaccines should be reasonable CD8 responses after the ad vectors. And we are now looking at some data in our own lab and what's also been published. The Pfizer vaccine gives reasonably good CD8 responses also. Less data out there on the Moderna.
Okay.
I don't know if that's your question or not, but essentially it's
thank you.
trying to not be biased, but just looking at the data.
So what are the odds of this mRNA coming and integrating in our genome as a transposon that anti-vaxxers are promoting everywhere?
Where is the data for it?
I think
it's all speculation.
No, I think it's close to zero. I think the mechanism doesn't exist. It's zero.
Exactly.
Scientifically. Just
Okay.
the machinery is not there for the integration.
RNA is so unstable. I mean, for that to integrate and propagate as a transposon.
No, but more than that, the enzymes are not there.
Yeah.
Exactly.
Okay. So Rama, you wanted to ask 4 questions. You are given 1 question now, and maybe you can come ask your most important question now and come back later.
Sure, sounds good. Okay, so Dr. Ahmed, I wanted to ask you, can you speculate why, like from the immune system point of view, SARS-CoV-2 elicits neutralizing antibodies quickly, but Ebola does not? So that's an advantage, right? Getting neutralizing antibodies quickly.
Yes, it is. Yeah, I know. Yeah, that's why I was very surprised, Rama, that binding antibody you get very quickly for any infection, also any primary infection. Also, isotype switching can happen very quickly. But neutralizing antibody is not that, as you know, from trial studies, that Ebola neutralizing antibody came up very slowly, even the binding antibodies were there. And with many other infections, this is the case. But here they came up very quickly. Where you do see antibodies coming up reasonably well, actually, is your favorite virus, yellow fever. You do get good neutralizing antibody. So I think it's very virus dependent, perhaps on the epitopes, which are very immunogenic. But this has been, I think, the best thing about this. If there's anything good about this horrible virus, is the fact that this S protein is immunogenic and induces good neutralizing antibody. That's why I think every vaccine that's been tested has had some protective effect. Some are more than others, but almost all of them protect from severe disease, and up to, some of them, it's 50% from infection, some more, but from severe disease, pretty much all of them are protecting. And I think because they're all inducing at least some level of neutralizing. And the virus is indeed neutralizable, indeed.
You're not doing any mouse experiments to address this? That's one question. The second thing is, yellow fever should be your favorite virus too. Yellow fever vaccine should be yours too.
Yeah, no, we in our lab we won't be, but Mehul and many other people are doing a lot of mouse experiments, especially in terms of the variants. I think we need to see which antibodies protect against, which vaccines protect against the variants. So Mehul and probably 100 other labs in the country are using mouse models to address those critical questions.
Okay, Kasturi Mitra.
Hi, I really enjoyed your talk. Wonderful. My question is about the trajectory of the virus. I'm interested in your take about what do you think the trajectory of the virus will be, and what will be the contribution of natural infections versus vaccines? And of course, there is the complication of the variants there, if you could comment.
Yeah. You mean trajectory of the virus in terms of
in terms of it will get so it will become like a flu virus in the population, and probably we'll have to take vaccines every year or every season. And will we ever be able to be completely immune, that we won't have to ever take the vaccines if a population of herd immunity?
Yeah, that's a very important public health question that I think everyone is asking. We of course don't have definitive answers on that question, but my take on that will be that we have to learn to live with SARS-CoV-2. I don't think it's going away. it spreads so efficiently and so dramatically in, all over the world that we'll be dealing with this. In the best case scenario, it might become like another coronavirus, but I don't think that will happen. This is much more pathogenic than the common coronaviruses. Its receptor binding is different. So I think that we will be dealing with it. Then the question becomes, what will happen in terms of vaccines? Hard to predict with certainty, but my guess would be that we will be getting boosted. How often we get them, I don't know. And that will relate to the variants. How much is this virus changing? And that relates to what was driving the selection. So here I tell you a little bit of my thoughts on the variants that have currently emerged. So where have these variants emerged? Have they emerged due to immune selection or due to post-adaptation at this stage? My feeling is that at this stage, many of the variants are more post-adaptation, better binding to the receptors, more transmissibility. All of these will give that virus an advantage. Some of those post-adaptation mutations will result in escape for antibody, as we see it. But whether it really was driven by antibody or by host adaptation is unclear. But my guess is that many of these actually was post-adaptation, because I think these were happening in naive individuals. But now, as we move forward with more people who are vaccinated, more people who are immune by vaccines, here I think we will see selection, immune selection, because they will now be trying to infect people who have some degree of pre-existing immunity. So I think this is going to be a very interesting thing to follow. How much immune selection is resulting in variants? If that continues, then I think we will be getting vaccines. It will kind of become a little bit like influenza. I don't know if we'll get it every year. I hope not, but it might be. Every few years, it might be needed. But I think the two bottom line messages that we'll be living with this virus, and if it continues to adapt and change due to immune selection pressures, I think we'll be getting additional booster vaccines.
So, Rafi, in that connection, one apprehension is, at least in India, is that rampant use of plasma therapy without first testing for the quality of antibodies in plasma could also have driven some of the selection.
You can't rule those things out, can't be ruled out at this stage. I don't know how widely it was used, but certainly.
Oh, very widely, very widely. In the first,
absolutely,
the first wave, very widely.
Yeah, it could, it easily could. There are many sources of variants emerging. One is what you just said, that could happen. The other possibility is that people who are immunocompromised,
right,
for some reason or the other, but not like fully immunocompromised that they make no immune responses. Okay? So there you will get persistence and long-term shedding. And there could be variants selected in those individuals.
Yeah, so what about people who are getting steroids to take care of inflammation while the virus is still there? I mean, wouldn't that also?
Well, that you will know, right? You know that if it turns out that they've controlled the infection, then I wouldn't be concerned. But if they are continuing to shed long term, then I think it would be a concern. Yeah. And there are some rare examples of this. I heard a talk from a group in South Africa, in Durban, where they have an HIV infected woman. She's on antiretrovirals, but low CD4 count, and that she's been shedding, apparently being PCR positive for COVID for 60 days or 90 days. And there are variants coming up in her. There's an N of 1, but at least shows you that that can happen if you have some long-term, some persistence of the antigen, of the virus.
Anindita Banerjee, please go ahead.
Yes, sir. Thank you for the wonderful talk. I was just thinking about the immune selection, which you were talking recently about the spike variants. You said that it is more on the host adaptation. So I was just thinking that would these emerging spike variants might have any difference in the immunological profile, like the cytokine surge. So you just
Yeah,
said.
there could be other mutations that happen in these variants that we're not looking at. We're focusing right now on the S protein correctly because that's where the antibody binds. But there could be virulence mutations happening in other parts of the genomes. There could be some changes to T cell epitopes. I think this is a very open area at the moment. And again, these steroids have so there's a lot of questions that I think we'll be addressing in the coming years. No definitive answers on any of these important questions.
Yeah, so the spike variants might elicit various immunological profiles based on other host parameters also, or their history of previous history of infection or the medical profiles. You were trying to say that?
No, I wasn't saying we don't know that, but what I was saying is that that there are many different ways that you could be selecting for variant. One, as was pointed out by Shahid, could have been plasma therapy was done with suboptimal sera that could have resulted in it. Certainly, people who are immunocompromised and are shedding virus for a long time, that means there's immune pressure on the virus, so you could get it that way. And now, as more and more people are getting vaccinated and are recovering from acute infection, there would clearly be an advantage for a mutant to infect them. So I think this is a changing kind of landscape that we'll have to be following very carefully.
Thank you, sir.
Jayanta, go ahead. I know you have a number of questions in the chat box and you've been waiting very patiently. Go ahead. Or maybe Jayanta is no longer there.
Hello.
Yeah, Jayanta,
Hello.
are you there?
Thank you for a great talk, sir. So I would just like to know, was there
Hello?
any effort to...
Hello. Who is this?
Hello. Sir, I'm Nitin. I'm working with Dr.
Nitin,
Jayanta.
just wait. I'm taking people as they ask questions.
Sorry.
So please wait your turn. Can I ask Iyad Ali?
Oh, thank you, sir. My question is why children are not advised to take the vaccine, although in most of the cases they were the asymptomatic carriers?
Yeah, a very good question. I think that's driven by the fact that very few children showed clinical symptoms. I think that was the main reason for focusing first on the elderly, where most of the deaths were happening, and then going down to the younger adults. So it's not that children should not get this vaccine. I think in the next years to come, I think children will also be getting this vaccine. As you know, in the US, it's gone down to 16 very quickly. Then it went down to now 12-year-olds are getting it. So 12 to 16-year-olds are getting it. Clinical trials have already started by Moderna going down to 5 years. So I think we'll be moving in that direction. The priority initially was vaccinating people who are dying and getting severely ill, and then moving down. It will become a childhood vaccine, I think, in the long run.
Okay, Dr. Omerlin, go ahead. Dr. Omerlin, are you there? I guess not. Anupriya?
Thank you for the talk, sir. So I wanted to ask, because we are using so many drugs, so multiple drug resistance is a common thing which we'll be facing, or we are facing also. So what effect it is giving to the vaccines which we are taking, or is it like there is no comparison between the two?
I mean, I'm not sure what you mean by drug resistance, because There are no antivirals being taken, so there's no antiviral resistance that way. So are you thinking of the drugs interfering with vaccine responses? Is that what you're thinking of?
Yes, sir. Because if we take
Yeah,
an
I think if
example of
somebody
a
is
patient
on,
who's been COVID affected.
yeah, I think if somebody is on some certain immunosuppressants and taking the vaccine, it will, depending on what they're taking and what the dosage is of those drugs, it could impact vaccine responses. Again, some of these studies are going to come out now because specialized patients are being looked at. But just a healthy individual taking steroids on their own, I don't know what the impact there would be. Probably not a good idea to be on steroids when you're taking the vaccine. I would recommend that they get off, if they're healthy and really don't need the steroids, to at least get off the steroids while they're taking the vaccine. I'm sure they will survive without the steroids.
Nitin, go ahead. Go ahead, please. Thank you for waiting. Go ahead.
Hello. Yes, I wanted to know, was there any effort to isolate monoclonal neutralizing antibodies from these cohorts? And what is your take on the utility of these neutralizing antibodies against the fight with the COVID?
Yeah, Nitin, yes. Literally thousands, if not tens of thousands of monoclonals have been made by people all over the United States and other places, including Jens Ramat at Emory and many people. But what we are most proud of is that our group in India, Anmol and Murali and the group there, they have made over 200 monoclonals from the patient cohorts in India. These were samples provided by the ICMR to our lab at ICGP. And many of those are actually quite good neutralizing antibodies. And we are currently testing if they neutralize the variants. Potentially could be used for treatment.
Well, we've come to the last question. Subhash Mehdi, go ahead and ask your question. Thank you.
Sir,
yeah, go ahead, Subhash. Go ahead.
Sir, at the current scenario, there are many brand of vaccine that has been given. In our country, we have Covishield and Covaxin that has been given now. So, as we all know that the antibody to the vaccine it does not last for longer duration. So later on, can we get other brand? Means this time Covishield, if we get, I don't know whether it will be possible or not to get other brand of vaccine later on.
Yeah, I partly addressed that question before. I think you could. I mean, scientifically, you could do that if you've gotten, let's say, if you've gotten Covaxin and you want to take Covishield and that's available. Yes, I think it will. It certainly will not hurt. It's unlikely. I think it would be fine. Also, you mentioned that the antibodies will not last. We don't know that yet. So I think that data needs to be determined. That what is the half-life of antibody responses after Covaxin versus Covishield. Not that difficult to do. I think that kind of study that we did can be done anywhere. You enroll the vaccinees and do longitudinal bleeds and just do neutralization or binding assays. So I think these studies need to be done in India, looking at longevity. And I've been telling many people at DBT and ICMR to try and conduct these studies. Enroll the vaccinees and follow them longitudinally for up to a year. And the study design doesn't have to be too complicated. You can just take one bleed every 3 months. You get like four time points over a 1 year period and see what the decay curve look like. It's very important to see longevity data with the Indian vaccines in the Indian population, and especially if the antibodies there are neutralizing the existing variants. This is essential to do in India. Otherwise, there'll be a lot of speculation and without any hard data. And these kind of studies should be done not by just one group. They should be done by multiple places.
Rafi, there's one last question coming from Akanksha Chaturvedi. Akanksha, go ahead.
Sure. Thank you, Dr. Jamil. Thank you, Dr. Ahmed, for the wonderful talk. I have a question. So why do we see that the most children remain asymptomatic? Your data showed a lot of cross-reactivity to the coronavirus, other family of coronavirus. So that could be the case, or because they have probably much more widely reactive clones for the B cells. I don't know about T cells too. And then what has changed in the second wave in India? Because now we see children getting a lot of symptoms as well.
Yeah. Let me take that cross-reactivity question first. So we saw really no cross-reactivity at all in terms of antibody responses. That's the only thing we looked at between the alpha coronaviruses and SARS-CoV-2. There was absolutely no increase in binding antibody to the two alpha coronaviruses. We did see something to the two beta coronaviruses, but again, the response was, we could pick it up because we showed you that pre-pandemic, and that one is not that great. Also, work that we have done and many others have done have shown that the B cells that you isolate and characterize early on, that is within the first month after vaccination, are all not coming from naive B cells. That is, if you isolate, use spike protein to isolate the spike-specific antibody, make monoclonals or do deep sequencing of those B cells, and look at level of somatic hypermutation, is hardly any. So most of even though we do have some cross-reactive B cells are there, the majority of the response is recruiting the naive B cells, because I think the affinity of those cells is much higher. So it's not that you cannot get cross-reactive responses, but in the infected people, they seem to be contributing to not such a great extent. Plus the data that we have in terms of susceptibility of the population. Common coronavirus seropositivity is close to 100% in most places of the world. But people are still so highly susceptible. The variability in different individuals, why some get more sick, some get less sick, whether that's related to what we had from before, I think it's an open question, no clear data.
Okay, thank you so much, Rafi. I know you've been talking for about 2 hours now.
I was just looking and it is almost noon. Yeah.
Yeah. So,
but wonderful audience. Really great. I want to thank all of you. Really great questions. Made me think also about a lot of things.
Thank you very much. And thank you everyone who's still here. And thank you for being here. Thank you for asking the wonderful questions. And do join us in future editions of the Distinguished Lecture Series, which will be announced. We do roughly one every month. So thank you everyone, and thank you again, Rafi.
Okay, thanks everyone. Bye-bye.
Bye.